Good morning, my YouTube viewers. Crystal here. I decided that I wanted to make a video for you this morning because I've been working on something called PCA. I think that's called Principal Component Analysis, but I'm not sure exactly. Whenever I do my videos, whenever I do my blog post, I'll get the term accurate on my blog post. And I'll also leave the link for you in my blog post to my GitHub account if you want to look at the program that I've written. But basically, the reason why I decided to use the E.Coli dataset is because it is highly imbalanced. It has, I believe, 8 classes, and I've not been able to get all 8 classes represented when I carry out predictions. And I've used a number of models. The most recent models that I used were CatBoost and Random Forest, and one class is always left out. So as a result of that, I thought, well, it's going to be a good idea to actually look at this. So we decided that we'd use PCA, which is an sklearn. It's called Principal. It's just called PCA. And whenever I do my blog post, I'll tell you a little bit more about it because I'll look it up and research my work. But what we'll do here is we're going to just read about the E.Coli dataset. So the dataset describes the problem of classifying E.Coli proteins using their amino acid sequences in their cell localization sites. That is, predicting how a protein will bind to the cell based on the chemical composition of the protein before it is folded. The dataset is created by Kenta Nakai and was developed into its current form by Paul Horton and Kenta Nakai in their 1996 paper titled A Probabilistic Classification System for Predicting the Cellular Localization Sites of Proteins. In it, they achieved a classification accuracy of 81%. And I know that when I used CatBoost and Random Forest, I achieved a higher accuracy than that, but I could not get all 8 of the types of E.Coli covered in the prediction, and I've not been able to do that. So the dataset is comprised of 336 examples of E.Coli proteins, and each example is described using 7 input variables calculated from the protein's amino acid sequence. And then it gives you a whole bunch of information. It gives you the amino acid sequences, and then it gives you the 8 classes, which I won't talk about that. You can read that. And basically, what I wanted to say is that, I've worked on this E.Coli set for quite a while, and I've never been able to get all 8 of the types of E.Coli predicted properly in my models, which is why I thought, well, this is going to be a good set to use PCA on so we can do an analysis. Now, PCA is a type of unsupervised learning. But the first thing I did was I created my program using Google Colab. And the good thing about Google Colab is that it's got all of the libraries already installed. Well, most of the libraries already installed. So all you have to do is import the libraries. So that's all we had to do, was just import the libraries. And then, so now I loaded my data set. The data set did not have a header, so I created the columns of MCG, GBH, LIP, CHG, AAC, ALM1, and ALM2, and the result. So those are your columns. So we checked to see if there were any null values, and no, there were no null values. So now what we did was we counted it. We counted to see how many examples we had in each class. So you see class IMS, you only had 2, IML, you only had 2, and OML, you only had 5. So that's going to be where we're going to have difficulty, because we're going to have difficulty getting those represented when we carry out our predictions. So we set our features. Feature equals trained columns, and so these are your features. So now what we've done is we've defined X, and the reason why, because this is unsupervised learning, we don't have a Y value, and because we don't have a Y value, we only have to define our X value. So X equals train dot drop result, axis equals 1 values, X equals standard scalar dot fit transform X, X equals shape. So we put the values into a scalar, and the reason why is because all the values have to be scaled. So we did a shape, and it's 335 over 7, which is interesting because the description of the data set said that there were 336, but I counted in all of my experiments, it says it's only 335. So there's one row missing somewhere, and I don't know how it was missing, but it wasn't. So now what we do is we calculate the mean value and the standard value. So the mean value is 1.7877, etc., to the negative 16th. And then the standard value is 1. So now what we do is we set up a variable called feature columns, and feature columns equals feature plus string I, for I in range, X dot shape 1. So basically, so we go to feature 0 to 6. So that's our 7 columns. And now what we do is we normalize the data. Normalize is something that you have to do in order to use PCA. So normalize equals PD dot data frame X columns equals feet columns normalized. So now you can see what all the values are as they are normalized. Now we have created our model, and from sklearn decomposition import PCA. PCA coli equals PCA in components equals 2. Principal components coli equals PCA coli fit transform X. So I think you can have as many in components as you want, but you have to define them. You have to define all the components that you want. So in this instance, we're only having 2 components. So we're having principal component 1 and principal component 2. And since I'm new at this and I've never done this before, I've never seen any with principal components 3, but I guess I can try that out sometime if I wanted to. So now what we do is we print explained variation per principal component. So dot format PCA coli explained variance ratio. So the explained variation per principal component is 0.31513592 and 0.2090622. So that's your explanation. So now what we're going to do is we're going to plot it on a graph. And so this is the formula to plot all of these dots on the graph. So the target is CP, IM, PP, IMU, OM, OML, IML, IMS, and those are the 7 or 8 classes in the target column, in the result column. In colors, you want every class to have its own colors. So I had to make some stuff up, So we've got red, green, blue, black, yellow, gray, purple, and then I picked maroon. So this is the chart that comes out. Principal component analysis of E. coli dataset. And so you've got all the red scattered together, and you've got the green scattered together, you've got the blue scattered together, you've got the black scattered together, you've got the yellow scattered together, you've got the gray scattered together, you've got the purple scattered together, and you've got the maroon scattered together. And I think that is one of the reasons why it's so difficult to work on this data set, partly because it is imbalanced, and partly because all of the examples are sort of like scattered together, and it's difficult to partition them. So I noticed that, like on the validation set, there was only like one, like on category 2 and category 3, there were only one example of category 1, 2, and category 3. So I think that's one of the reasons why it's so difficult to work on this data set, is because all of the different values happen to be scattered together. And what I'll have to do is I'll have to do some more work on principal component analysis. I am new to this, but I know this is a type of unsupervised learning, and what it does is it allows you to detect outliers, it allows you to detect fraud. So we've got an outlier here, over here on the purple, which IML is an outlier. But it's just a way to detect outliers, a way to detect anomalies, and a way to actually look at your data without performing a prediction. So that concludes this video on the E. coli dataset. If you like my video, please like, subscribe, and share. If you like the work I do and want to leave a donation, I've got my email address to my PayPal account in the description box down below. And thank you so much for watching my video, and I look forward to making more videos for you.